Good afternoon. My name is Eliza and I'm from Nara Institute of Science and Technology, Japan. So it is a great pleasure to be here to present my work to you today entitled Recognition of Microbes and Host Damage Associated Patterns Trigger Salt Stress Tolerance in Arabidopsis thaliana. So we know that in nature, plants coexist with microbes under the influence of the environment. And this three-way interaction will determine the plant's fate and susceptibility to infections. So a lot of people ask, how do the environment influence the plant-microbes interactions? But another way to look at this three-way interaction is to ask how the microbes actually influence the plant's adaptation to the environment. And that is exactly what I'm interested in, especially when it comes to the plant's defense system. So earlier, Jessica made a very nice introduction on PTI, and then Paul and Nico introduced what are patterns and receptors. So I'll go through this really quickly. So the plant's pattern-triggered immunity starts with the recognition of patterns, such as bacterial patterns, FLAG22, ELF18, and plant-derived patterns, such as PEP. And these patterns are recognized by pattern recognition receptors, or PRR, and together with the co-receptor BAK1 will lead to the activation of defense genes. So very interestingly, we found that these defense-responsive genes are also induced under salt stress. So we took PEP2 and ELF18 inducible genes and looked at the expressions of these genes under salt stress. And we found a very high overlap between these two gene sets. Like we see over here, the precursors and also the PEP receptors are induced under salt stress. So this indicates that PTI components may play a role in salt stress responses. So to validate this expression analysis, we traced and detected the induction of ProPEP3 in the roots of Arabidopsis thaliana when they are placed under salt stress. So these PEP peptides will naturally be secreted out of the cell, and indeed we did manage to detect the presence of these ProPEP3 in the growth medium in which the plants were grown under salt stress. So to further validate or to ask how PEPR is involved in acquired osmotic tolerance, we grew the plants normally and then we placed it under mild salt stress before subjecting them to osmotic stress. And we found that the plants that were pre-treated with mild salt stress could actually tolerate osmotic stress better. And this is in a manner that is dependent on PEP receptors. As you can see over here, the receptor mutants have very low chlorophyll content compared to wild type, and especially lower compared to PEPR overexpressing lines. So this tells us that acquired osmotic tolerance is dependent on PEPRs. So consistently in an assay that I've developed, we see that PEP1 application enhances salt stress tolerance. So in this assay, 4 days old plants were treated with PEP1 before they were transferred to medium containing high salt. So as you can see over here, plants that were treated with PEP peptides have higher number of survivors under salt stress compared to plants that were mock treated. And this is again dependent on PEP receptors. As you can see here, the double receptor mutants are compromised in PEP-induced salt tolerance. And this phenomena is not just specific to PEP1, as application with PEP2, 3, and 4 all induces enhanced salt tolerance. We also tested the we also tested whether or not MAMPs, bacterial MAMPs, such as FLAG22 and ELF18, could induce salt tolerance, and indeed they could. So as you can see here, pre-treatment of plants with FLAG22 and ELF18 induces enhanced tolerance. And this is again dependent on their pattern recognition receptors. As you can see here, FLS2 and EFR double mutant are incapable of induced salt tolerance. We therefore call this phenomena the pattern-triggered salt tolerance, or PTST. Several studies have also reported very similar phenomena. So just very recently, Nakaminami's group reported that overexpressing PEP3 or pre-treating plants with PEP3 actually induces salt tolerance, and they also reported that PEPR1 is involved in this phenomena. In this work, however, we found that PEP1, 2, 3, and 4 actually induces salt tolerance, and both PEPR1 and 2 has a role in this phenomena. And then 2 other independent studies reported that fungal chitin and SIRT1 do contribute to salt tolerance, but however, in this study, we found that pre-treating plants with chitin do not induce salt tolerance. So the distinctions in all these studies point to the diversity and the complexity of this cross-tolerance. But very importantly, none of these studies reported the mechanisms underlying this MAMP or DAMP-triggered salt tolerance. So therefore, we are very interested in pursuing this question. So we first tested whether or not abscisic acid is needed in this PTST, mainly because abscisic acid plays a major role in abiotic stress tolerance. So we used 2 different mutants, the ABA biosynthesis mutant and ABA responsive transcription factor triple mutants. And as you can see here, in both scenarios, these mutants show wild-type phenotype. So this tells us that abscisic acid is not required for PEP1-induced salt tolerance. However, other PTI components are required. So just to recap, BAK1 is the co-receptor for PRRs. And then we have 2 mutants here. BAK1/4 is a null mutant that can retain PTI signaling. BAK1/5 is a mutant that is specifically impaired in immune signaling. And as you can see here, in BAK1/4, this mutant managed to retain PEP-induced salt tolerance. However, the immune signaling mutant could not induce enhanced salt tolerance. So this tells us that BAK1-mediated immune signaling is needed for PTST. So similarly, we see that RBOHD mutant that is incapable of defense-associated ROS burst could not or have attenuated PTSD. So once again, both of these PTI components, BAK1 and RBOHD, are required for PEP-induced salt tolerance. Next, we wanted to see how PEP application could influence the transcriptome of the plants under salt stress. So we performed RNA sequencing using wild-type and PEPR double mutants that are treated with PEP1 without salt stress, with 3 and 24 hours of salt stress. Our RNA sequencing analysis revealed close to 2,000 genes that are dependent on PEP1. And these genes are clustered into this very complicated heat map over here. And they are clustered into 21 different clusters. And some of the clusters are very interesting. For example, this cluster that I've boxed in black over here shows PEP1 enhanced salt-inducible expression. So taking these DEGs, we perform an in silico analysis to look at whether or not there are any overrepresentation of cis elements. And we found that the WRKY transcription factor binding sites are enriched in the upstream promoter regions of these genes. We then choose to focus on WRKY 18 and 40, mainly because there's extensive studies and reports that they directly bind and negatively regulate PTI-inducible genes. And another reason is because WRKY 1840 have very similar co-expression patterns. So in order to test whether or not WRKY 1840 contribute to PEP1-induced salt tolerance, we use 2 different alleles of WRKY 1840 double mutant. And as you can see here, pre-treating these mutants with PEP1 did not enhance salt tolerance. Okay, all right. So this tells us that WRKY 1840 contribute to PEP-induced salt tolerance. So we then perform qPCR analysis on a marker gene that is selected from the heat map. So this marker gene, PTR3, as you can see here, is de-repressed, the expression of which is de-repressed in WRKY 1840 double mutant under salt stress. So this tells us that WRKY 1840 positively regulates PEP-induced salt tolerance. Now the next question is, how exactly do WRKY 1840 regulate PTST? So in an earlier slide, I've shown that acquired osmo-tolerance is dependent on PEPR. So in the very same acquired osmo-tolerant assay, our previous study shows that EDS1-mediated defense protein, EDS1, part 4, and this NLR aqueous, the loss of which leads to enhanced osmotic tolerance. So this tells us that EDS1 defense inactivation confers osmotic tolerance. So to make this easier to understand, so far, I hope I've convinced you that PRR defense activation induces salt tolerance. However, in our previous study, we showed that EDS1 mediated defense inactivation confers osmotic tolerance. So how do we explain the apparent discrepancies between these two defense-mediated osmotic or salt tolerance? So we hypothesize that WRKY 1840 may be the possible link. So supporting our hypothesis, there are several studies that reported negative relationship between WRKY 1840 and EDS1. So as you can see here, in WRKY 1840 mutant, this mutant shows enhanced fungal resistance. But such enhancement was cancelled with the simultaneous loss of EDS1. So this tells us that WRKY 1840 suppresses EDS1 function. Now using the similar approach, we tested the relationship between WRKY 1840 and EDS1 during PEP-induced salt tolerance. So like I've shown you earlier, in WRKY 1840 double mutant, the PEP-induced salt tolerance is compromised. However, with the simultaneous loss of EDS1, this compromised phenotype reverted back to wild-type phenotype. So this tells us that WRKY 1840 suppresses EDS1 function in order to confer salt tolerance. So in good accordance, when we perform qPCR analysis of EDS1, we see that EDS1 expression is de-repressed in WRKY 1840 during PTST. So collectively, our data tell us that MAMP recognition by PRR activates WRKY 1840, which then suppresses EDS1 function in order to confer salt tolerance. So since all these assays were conducted under sterile condition, our next question is, could bacterial sensing lead to salt stress tolerance? So in order to test this out, I've sprayed inoculated seedlings with different strains of Pseudomonas syringae DC3000 before subjecting them to salt stress. The strains that we used were PTI-inducing strain, HARP-S, and two different ETI-inducing strain, avian RPM1 and avian RPM4. So as you can see over here, the PTI-inducing strain, HARP-S, is capable of conferring salt tolerance. However, the two ETI-inducing strains were no different from mock-treated samples. So this tells us that PTI is greater than ETI in conferring salt tolerance. Now, this non-pathogenic bacteria-induced salt tolerance is also dependent or requires PRR complexes. As you can see here, this BAK1 BKK1 double mutant, this mutant is incapable of full activation of PTI signaling. And as you can see here, this mutant is incapable of inducing salt tolerance. And what is interesting is that the loss of just two pattern recognition receptors, FLS2 and EFR, is already sufficient to attenuate bacterium-induced salt tolerance. So this points to the significance of pattern recognition receptors when it comes to bacterium-induced salt tolerance. So consistent with the sterile assay, we see that bacterium-induced salt tolerance also requires the function of WRKY 18 and 40. Well, once again, we see that pre-treating this mutant with bacteria did not result in higher number of survivors under salt stress. Now, we repeated the assay with a different non-pathogenic bacterium, Pseudomonas fluorescens, and this strengthened our claim that non-pathogenic bacterium could induce salt tolerance. But what is very interesting is that heat-killed PFO here, could also induce salt stress tolerance. So this shows that the mere perception of MAMPs is sufficient to induce salt tolerance. And importantly, we see that in the double mutant, this immunocompromised mutant, the bacterial growth is not significantly different from wild type. So this tells that the defects in this immune signaling mutant is not, the PTST defects, is not due to bacterial overgrowth. So therefore, in conclusion, We have introduced pattern-triggered salt tolerance, or PTST, whereby MAMP or DAMP recognition by PRR leads to salt tolerance. And in this work, we also identified WRKY 18 and 40 as the potential switch to either favor PRR defense activation or EDS1 mediated defense activation to confer salt or stress tolerance. And finally, we've presented evidence that non-pathogenic bacteria could also confer salt tolerance in a manner that is dependent on pattern recognition receptors. So with that, I end my presentation with a special thank you to my PI, Yusuke Saijo, who couldn't make it to this meeting. But this is his contact information if you are interested to talk to him. Our collaborators, and also, of course, our fundings. So with that, thank you very much.